Good morning, everyone. We're going to go ahead and get started. I'm Sangeeta Bhatia. I'm one of the members of the Koch Institute and excited to be moving into the new building in November. I'm going to be your session moderator this morning. The session is on detecting and modeling cancer. So to get started, I'm going to be introducing Joe DeSimone. He's our first speaker. He's a professor of chemistry. He received his PhD in chemistry from Virginia Polytech under the guidance of James McGrath. He joined the Faculty of Chemistry at University of North Carolina, Chapel Hill in 1990, where he's been ever since. He's currently the Chancellor's Eminent Professor of Chemistry and the founding director of the Institute for Nanomedicine there. His work spans many areas of nanomedicine, most recently the systematic study of the impact of nanoparticle shape on biological interactions and many other areas. He's the co-founder of Liquidia Technologies, which uses a templating method to manufacture engineered nanoparticles for life sciences and material applications. Among his many, many awards are the Lemelson MIT Prize, awarded here, given for inventors dedicated to improving our world through technological invention and innovation, the NIH Director's Pioneer Award. He's also a fellow of the American Academy of Arts and Sciences. He's a friend to the Koch Institute, as he's sat on our advisory board since last year, so we'll be happily seeing much more of him in the years to come. The title of his talk today is Nanofabrication Technologies for the Production of Vaccines and Therapeutics. Joe, welcome.
Good morning, everyone. Thank you for inviting me. It's terrific to be back and see a lot of great friends, and want to thank Tyler and Bob for the invitation back. My relationship with Bob has been for some time. This is a fully bioabsorbable stent that we developed with a company about 8 years ago, and it was great to work closely with him. That's in about 1,100 people today in an international clinical trial, so it's been a great relationship to continue working with Bob. But this morning I want to tell you about a story related to nanoparticles, and in particular our approach to this. You know, there's a lot of effort in making nanomaterials, and I think if you think about nanomaterials, there's basically two general approaches for the fabrication of nanomaterials. There's sort of the bottom-up self-assembly, atoms into clusters of atoms like quantum dots, or molecules into self-assembled supermolecular structures like micelles and liposomes. This has pretty much been dominated by the world of chemists of different types. And then there's this whole other class of nanomanufacturing that's more the domain of the semiconductor industry, these so-called top-down lithographic techniques, which have been used to drive the fabrication of different types of microelectronic devices and microelectromechanical type systems. And my group is interested in trying to tap these types of techniques for making organic nanoparticles useful in a variety of different biological systems. Eric Lander mentioned yesterday about Moore's Law, and it's interesting to think about Moore's Law in a biological context. Back in the early 70s, when folks were buying computers, the chip size was about the same as it is today, about a square centimeter, and one could cram about 2,000 transistors onto that chip because the minimum feature size of these transistors was about the size of a single cell, about 10 microns. 40 years later, you buy a computer, the chip is about the same size, but now the minimum feature size is on the order of the size of a virus particle. And you're able to achieve about multiple billions of these on the same relative area. And now we're talking about advanced precision technologies that are in the range for starting to probe biological systems. And this is a context that we want to apply. But obviously, these lithographic techniques are usually used for metals and metal oxides and are not conducive to biological systems. So my laboratory has pioneered a focus on translating these technologies into organic materials. And we developed a technique we call PRINT, particle replication and non-wetting templates. And basically what we do is we start with a standard silicon wafer, just like the standard lithographic techniques. We etch patterns into that wafer, and these are 100 nanometer features that are physically etched into the silicon wafer. We pour some different types of polymers on the surface of this wafer. This is a liquid fluoropolymer that has a positive spreading coefficient on just about all surfaces. It will wet every nook and cranny of this wafer at the nanoscale. It's designed to be photochemically cured in tens of seconds. So once it spreads and wets, we shine a light on it, and we peel off a membrane that now is basically a mold derived from that master template. But this mold is made out of a material that's got very similar characteristics to Teflon. It's a very low surface energy material. And what we typically do is take another material, another sheet or film, if you will, that has and I'm showing you here in red, a liquid that's going to be a precursor, a liquid or a polymer that's going to be a precursor to a therapeutic or a vaccine. And we basically spread that out on another sheet, and then we marry the two up, and we run these through a roller. And we do a film splitting technique, and we're able to uniquely fill these cavities without wetting the land area between the cavities. And now we have individual pools of liquids that we then solidify, and I use that term broadly. We convert that liquid to a particle, either through chemistry or a phase change. And now we have individual particles that when we then take that film, marry that up with another material, another film that has an adhesive on it, and this could be simply a water-soluble adhesive, run that through a roller, transfer all the particles out onto that film, and then we harvest those particles by simply running that film through another roller with a bead of water, typically, dissolves away that adhesive, have basically a dispersion of particles that we can lyophilize and have individual particles. So it's a templated manufacturing technique for making nanoparticles. And we've now developed this technique and pushed it all the way towards GMP clinical capability. This is a roll-to-roll machine at our company, Liquidia. We get thousands of linear feet of this film. We have a miniature version of this in our laboratory at the University of North Carolina. This is now GMP compliant, and it will support actually through phase 2 clinical trials that we're starting in vaccines, and I'll walk you through some of that. The particles that we fabricate now have the precision and uniformity of the microelectronics industry. You know, etching these features on a wafer, you can do it on this side of the wafer or this side of the wafer, and these techniques are highly reproducible. And so you end up with populations of extremely uniform particles. This is dynamic light scattering of our particles. So more often than not, this is a bell curve or sometimes even a mountain range of different maxima. We have, in fact, really monodispersed particles of controlled size and shape. And we've been able to fabricate a range of particles of different characteristic shapes. These are micron-scale particles where one could vary aspect ratio. We're very interested in inhalation products. I'm not going to talk about that today, but we can control particles in the micron size range, which is really important for deposition in the airway. These even have nanoscale etched structures on the surface of microscopic particles to prevent contact. These particles here are regio-specifically labeled because when the particles are in the mold, there's one face exposed. We can conjugate antibodies and peptides to that one surface. We can control modulus and a number of things. These are the particles on the nanometer scale. So these are tiny little discs that are just over 100 nanometers in diameter and 35 nanometers thick. So keep in mind, a red blood cell would be about 8,000 nanometers in diameter. These are getting on the length scale of individual macromolecular chain dimensions. These are the size of many liposomal products, and we're very interested in controlling the aspect ratio and making filamentous-like particles of controlled size and even flexibility. As you see, as we lift them off the surface, you can see they're deformable. And we have master templates where we can keep one dimension constant, and we can systematically vary the other dimension. And we're very interested in how objects like this interface both at the cellular level as well as delivered systemically through different routes of administration. These are examples of some of the particles we use most extensively in our laboratory for parenteral delivery. These are 80 nanometer by 320 nanometer particles. These are the largest particles that we can fabricate that can be sterilized by filtration as we start thinking about that. So that's the bridge. If you want to go bigger, you have to work aseptically. but these are filterable and you can sterilize. And I'll show you the range of chemistry is extraordinary. We can make particles out of any chemistries, ranging from antibodies to polymers to pure drugs. And so what we have the ability then is systematically controlling for the first time key fundamental particle parameters of size and shape from the nanometer scale to the micron scale, and then cover a range of chemistries that allows us to probe surface chemistry in a very defined way, because the particles are initially in an unaggregated state, and we have regio-specific labeling capabilities. We can control chemical composition, and we can control modulus. And so with this toolbox, with basically what we consider nanometer-sized tools with precise dimensions, we're interested in probing different biological systems.